Hello everyone. So for this particular video discussion, we are going to tackle the catabolism of carbohydrates with emphasis on cellular respiration. Um, from the last video discussion, no, you have been introduced formally to a nutritional metabolism. So let's formally discuss po, no, the individual metabolic pathways, starting with how we break down carbohydrates or how we catabolize carbohydrates. So, apil na diri actually ang katong three basic metabolic pathways that you probably know already, of course, especially katong duhan, no, katong glycolysis and Krebs cycle. So we are going to thoroughly discuss those pathways in this video discussion. Okay, for the summary or the content of this discussion. So let's start this discussion with cellular respiration, and then followed by fermentation or anaerobic respiration, Cori cycle, glycogenolysis, and catabolism of other monosaccharides. Okay, cellular respiration is actually an aerobic pathway. So that means um, in order for cellular respiration to occur, um, it needs oxygen. Okay, kung walay oxygen, then um, the the intermediates or the metabolites would be converted to other things. Okay, but so let's focus on condition that is aerobic or na ay oxygen, no. Okay, so excuse me. So cellular respiration is divided into three metabolic pathways. We have glycolysis, Krebs cycle, and oxidative phosphorylation through the electron transport chain. So actually, kaning ETC, no, it's not really a metabolic pathway. It is actually more of the structure that allows for the metabolic pathway, which is called oxidative phosphorylation. So the latter is the metabolic pathway, and the structure that allows it is the electron transport chain, which is found in the mitochondrion. So remember that the mitochondrion is the powerhouse of the cells. Okay, so nga naman gitawag siya nga powerhouse because diha actually gaka-produce ang daghan ng ATP. Diri actually no sa oxidative phosphorylation. Okay? So let's also discuss later on unsay pasabot aning substrate level phosphorylation and how does it differ from oxidative phosphorylation. So in this particular figure, no, these metabolic pathways are separated by an organelle. Of course, the Krebs cycle and ETC only occur in the mitochondrion. Okay, ang glycolysis is not actually situated in the mitochondrion, but it takes place in the cytosol or in the cytoplasm. Okay, or specifically in the fluid compartment of the cell, which is, you know, the cytosol. Okay, so okay, kanisad no kaning NADH ug FADH2 ato pud ni siya kanang atong i-deepen ang atong nahibaw-an ani. Of course, we know na that NADH is a derivative of niacin. Okay? And then FADH2 is derivative of vitamin B2, which is riboflavin. And you know already that there are electron carriers. So unsa ilahang function as an electron carrier? What is the purpose of them being an electron carrier? So mauna siya nga to ang mahibaw-an in the next slides. Okay, so have you ever wondered why exactly you need to breathe? What happens when you stop breathing? Yeah. So, yeah, nganong kinahanglan man gyud ta mag take in oxygen, nganong kinahanglan man gyud ta maginhawa, nganong there is a respiratory system that does that function. Okay, so actually the purpose is to sustain cellular respiration, because cellular respiration would not occur without oxygen. Mao na tong akong ingon ganina nga dapat na oxygen para ang cellular respiration mag-take place. Okay? So later you will fully realize nga nung what is really the function of breathing, what is really the purpose of breathing. Um, and that is cellular respiration. Okay. So this is the set of metabolic reactions and processes that take place in the cells of organisms to convert biochemical energy from nutrients into ATP and then release waste products. So, the bulk of cellular respiration occurs in the mitochondria. Okay? So, yeah, in this side, um, these are the heterotrophs. So, animals, fungi, many bacteria. Heterotrophs, it's because we depend on our source of nourishment from consuming other organisms. Okay, so kita, di ba, we are omnivores, so we consume plants and animals. So kana sila, kana mga foods nga makuha nato diha, of course you know that those foods contain the nutrients that we need, the proteins, fats, and carbohydrates that we need for our energy. Okay, yeah. And then remember guys that the product of cellular respiration, which I have already discussed in the last video discussion, no, nga makaproduce ta og carbon dioxide. Okay, so yeah, kaning carbon dioxide is, recall nga mauna siyang gamiton sa mga autotrophs or sa mga producers para makaproduce sila og carbohydrates, proteins, and fats. So the same carbon dioxide molecules that we emit, that we release, are the same carbon atoms that they are going to fix, that they are going to produce into the macronutrients. And as a consequence, pwede sila maka-trap og energy again from sunlight, and makaproduce sila og molecules, specifically carbohydrates, through the process called photosynthesis. Okay? And then, ang byproduct anang photosynthesis is oxygen. And that oxygen is what we need. Then, we release again carbon dioxide, and you know that the carbon dioxide is what plants and many autotrophs need. Okay, so it's just a cycle of chemicals, no? So, kung unsa ang atong gina-release, mao na ilang kinahanglan, o kung unsa ang gina-release sa autotrophs, mao na atong kinahanglan as the heterotrophs. Okay, so the producers or the autotrophs are plants, algae, and also many bacteria. Okay, so yeah, this is just a review kay na-discuss na nako ni in the last video discussion. Okay, so yeah, this is another representation of what I have discussed during katong introduction to metabolism. Okay. Okay. So yeah, di ba mao ni ang reaction sa photosynthesis. Okay, na ay ma-release ng oxygen, and then mao natong kinahanglan para makaproduce ng carbon dioxide, water, and mobalik na sad na sa autotrophs para makaproduce sila og another glucose molecules. So it's just a cycle. And you also have to remember the concepts of exergonic and endergonic reactions. Okay, so oxygen or no oxygen. So you know that with oxygen, that's aerobic, and no oxygen, that's anaerobic. So aerobic respiration requires oxygen, so this is really the cellular respiration. Anaerobic respiration is fermentation. Okay, it does not need oxygen. Remember guys that cellular respiration are different from anaerobic respiration. Okay, because of the presence of oxygen. So yeah, okay. It takes place in the mitochondria. So without oxygen is, again, it's called anaerobic, and it happens in the cytosol. Remember guys that among those three, no, si glycolysis ang ga-occur sa cytosol. So that means, would you say that glycolysis is an anaerobic reaction? If your answer is yes, then you're correct. Okay, so glycolysis can take place without oxygen. Okay, but that leads to my next question, no? Do Krebs cycle and oxidative phosphorylation via electron transport chain happen in the absence of oxygen? So the answer is no. Okay? So Krebs cycle and ETC can only operate under aerobic conditions. So unsa ganitong byproduct sa glycolysis, di ba? It's pyruvate. So kanang pyruvate kung naay oxygen, then musulod na siya sa mitochondria from the cytoplasm, because remember that glycolysis happens in the cytoplasm. So kung naay oxygen, then musulod si pyruvate to the mitochondria, then dito siya i-metabolize. Pero kung walay oxygen, okay, say for example mahitabo na sa ato kanang gadagan ta, okay, dili ta makakatch sa atong breath, magkulang ta og oxygen. So some of the pyruvate nga ma-form from glycolysis would not enter the mitochondria. Instead, since walay oxygen, ma-convert siya into other thing, and that is lactate. Okay, so that is just an overview kana ako ang gi ko ano kay we will deepen that things as we go along these slides. Okay, so aerobic respiration is actually a redox process, okay, because it requires oxygen. So aerobic respiration is used by most organisms. So nutrients, typically glucose, are catabolized to water and carbon dioxide, and energy is stored in ATP. So aerobic respiration is a redox and catabolic process. So the goal is actually to release the chemical energy that is stored in the food para i-capture sa ATP. Okay, so the other goal is to produce ATP so that makapture na tong energy nga na-harvest na to gikan sa foods. Okay? So, yeah. Kani nga ko ano nga yield sa okay. You would also expect na after discussing the three metabolic pathways involved in aerobic respiration, makabalo mo unsa ang pag-compute sa ATP nga ma-produce out of one glucose molecule. Okay? Sige. So, yeah. Glucose is oxidized to carbon dioxide and oxygen is reduced to water. So, makit-an pud ninyo no, unsaon pag-convert sa glucose to carbon dioxide. So, makit-an nato na along the several pathways, specifically in the Krebs cycle. Because wala pa yung na-produce sa carbon dioxide, right, sa glycolysis, diba? Naka-memorize naman mo sa glycolysis. So, mao ni ang nindot ani kay naka-memorize na mo daan sa glycolysis Krebs cycle. So, this wouldn't be so hard for you to understand. Okay. Okay. So, glucose is oxidized in the presence of oxygen. So, electrons leave the carbon atoms and combine with oxygen. So, si oxygen actually, wala siya sa glycolysis, wala sad siya sa Krebs cycle. Dito pa siya sa electron transport chain. Dito pa siya sa oxidative phosphorylation sa ETC or electron transport chain. So, this happens by a series of steps via NAD+ and an electron transport chain, which is located in the inner mitochondrial membrane. Okay, so later on, you will also review the structure of the mitochondrion para mas masabtan ni. So, yeah. So, that's why kinahanglan yun og oxygen. Okay? And you know that it's catabolic, so therefore, ang atong goal is, so the nature is oxidative. Okay? And you know that if electrons are removed from a molecule, then that process is oxidation. Okay? Ang bali niya is, di ba, reduction. Okay? So, reduction is kung mudawat ka, kung kani siya nga molecule mudawat og electron, then that molecule is being reduced. Okay? So, yeah, you know that things already kay mauman na to ang na gidiscuss naman sa dako na sa katong midterm. Okay, so the goal of respiration is to produce ATP. Nganong gitawag siyang respiration kay kinahanglan lagi og oxygen. And para makakuha tag oxygen, we have to use our lungs. Okay, that's actually the function of your respiratory system. Para mo deliver siya og oxygen to the cells. And unsaon man ang oxygen gamiton sa cellular respiration para makaproduce tag kanang energy out of the foods that we eat. So that's how the respiratory system and the digestive system cooperate, no, when it comes to their functions, because the digestive system provides us the macronutrients, the digested macronutrients, and the respiratory system provides us oxygen. So mag-converge na sila dito sa sulod sa cells. So ang oxygen, then mga nutrients, so para i-oxidize katong mga nutrients and makaproduce ta ultimately og energy, and i-trap na to siya sa ATP. Okay, energy is released from oxidation reaction in the form of electrons. So, nauna-naon sad ninyo guys ha. Unsa lugar kanang kuan, like unsa nga energy, unsa man gyud nga unsa nga entity ang ginakuha nato sa mga nutrients para makaproduce tag energy. It's actually electrons. So magkuha lang ta og electrons from the foods that we eat. So from the macronutrients, strip off lang sila og electrons. O kana nga electrons mao na ang gamiton sa electron transport chain. Og dito sa electron transport chain dito i-harvest ang energy. So later on mas ma-realize na ninyo, no. So then para kato nga mga electrons makasulod sa electron transport chain, kinahanglan og mga shuttle or mga electron carriers, the NADH and the FADH2. So katong mga NADH nga na-produce sa glycolysis, di ba? Kana siya nga NADH, moderate na siya og electron transport chain. O katong FADH2 nga na-produce sa Krebs cycle, ipadulong po to siya sa electron transport chain. Okay? Remember guys that NAD plus, this is the oxidized form. Kung butangan nimo nig electron, mahimong NADH. So that's why NAD plus is reduced in many steps of glycolysis and Krebs cycle. Then kani si NADH nga reduced form, i-oxidize na dito sa electron transport chain. So si NADH mag-donate og electrons sa electron transport chain. So si NADH mahimong oxidized NAD, mahimo na sad siya NAD plus. Okay, then kana sila kanang si NAD plus mobalik na siya sa glycolysis o Krebs cycle to receive another set of electrons. So kana siya nga mga electrons, no, gikuha na to sa glucose. Okay, so later on mas ma-realize na to na, no. Yeah, electron energy is converted to ATP at the electron transport chain. So cells are able actually naatay duha ka klase nga pagbuhat sa ATP. First is substrate level phosphorylation, second is oxidative phosphorylation. So substrate level phosphorylation involves the transferring of phosphate directly to ADP from another molecule. Okay, and then oxidative phosphorylation is the use of ATP synthase and energy derived from a proton gradient to make ATP. So kaning substrate level phosphorylation mao ni ang gakahitabo sa glycolysis and Krebs cycle. Remember, di ba na ay ma-produce nga ATP sa glycolysis? Di ba na atay pila to ka ATP atong na-produce sa glycolysis, di ba duha? Then net na siya. Okay? And then sa Krebs cycle na asad tayo na produce nga GTP. Kana siya nga GTP later on ma-convert ra na into ATP. Okay, so therefore naatay na produce nga ATP sa Krebs cycle nga duha, then sa glycolysis duha. So actually, how are those ATPs made? They are made via substrate level phosphorylation because directly giphosphorylate ang ADP. So when you say phosphorylate, dugangan siya og phosphate group ang ADP. So di ba kung magbutang kag og isa ka phosphate group sa ADP, mahimo siyang ATP. So that's why it's called substrate level, because in the level of the glycolysis and Krebs cycle, in the substrate level. Whereas kaning oxidative phosphorylation, mao ni siyang phosphorylation nga kinahanglan og oxygen, and this only happens in the electron transport chain. So oxidative phosphorylation happens in the ETC, while substrate level phosphorylation happens in glycolysis and Krebs cycle. kay kinahanglan mag og ATP synthase, and ATP synthase is located near the electron transport chain. Okay. Aerobic respiration, guys, I've told you ganina is it's 3, but naatay ikaupat, no? And that's actually the pre-Krebs. Okay, so before mag-Krebs or before mag-citric acid cycle, so the other name di ay sa Krebs kay TCA cycle and citric acid cycle. So TCA stands for tricarboxylic acid cycle. So later on ako i-explain nga nung gitawag siya nga tricarboxylic acid. Okay. So yeah, naa pa dapat ma-form nga acetyl-CoA. Okay? So, from pyruvate. So, remember guys that the byproduct of glycolysis is pyruvate. And si pyruvate, kinahanglan pa siya i-convert into acetyl-CoA, okay, through the process called as pyruvate oxidation. Kani guys, kaning tulo, may tabo ni siya sa mitochondrion. Then, ang glycolysis sa cytoplasm lang. So that's why substrate level phosphorylation ay ma-produce sa glycolysis. Krebs cycle, substrate level phosphorylation. But oxidative phosphorylation only happens in the oxidative phosphorylation. Okay, so the overview of metabolism looks like this. Okay, so ang atong example lang sa kay carbohydrates lang sa, and that is in the form of glucose. Sa kanipon nga discussion, no, ako pod i-discuss kung unsaon nato pag metabolize sa mga non-glucose carbohydrates like glycogen. Okay, that's a polysaccharide. Unsa pa? Fructose, galactose, mannose. So those are other sugars. So unsaon nato pagkuha og energy gikan nila. So ato na siya i-discuss later. But unsaon pud nato ni pag breakdown sa fats ug amino acids? Natay separate nga discussion ana sa the following days, no. So focus ato sa carbohydrates. So carbohydrates, so in the form of glucose. So yeah, ang electrons, magkuha tag electrons gikan sa glucose, then i-donate na siya kay NAD plus og FAD para unsa, para ma-reduce sila into NADH and FADH2. Then kani si NADH and FADH2 mao na ang kinahanglan sa ETC. So i-oxidize sila sa ETC para ma-produce, para ma-regenerate ang NAD plus ug FAD plus, which are the oxidized forms. Then para makabalik sila sa glycolysis o Krebs cycle. Okay? And ang carbon dioxide, guys, are actually made from pyruvate oxidation. That's stage 2. That's the stage 2. And then the citric acid cycle. So later on, may bawaan ninyo nung dito nga pathways na produce ang carbon dioxide. And oxygen actually enters not in the Krebs cycle and glycolysis, but it enters in the ETC. Kay dito si oxygen man ang final electron acceptor. So kaning mga electrons nga gikuha nato, gi-remove nato sa mga nutrients, si oxygen ang mag-receive sa final. So ang pinaka final gyud nga mag-receive sa oxygen ay sa electron is oxygen, then forming water. So later on makit-an po na ninyo. Okay. So this is the cytoplasm and this is your mitochondrion. Remember that these are cristae, di ba? Mga curves. Then like any other organelle, mitochondrion has also two membranes. The outer membrane and then the inner membrane. Then the space between the outer and inner membrane is called the intermembrane space. Okay? So ang ETC nimo actually na-locate siya sa, naka-embed siya sa inner mitochondrial membrane. Then ang Krebs cycle actually naa siya sa matrix. Okay? So mao ni ang sulod sa inner mitochondrial membrane. So sa Krebs cycle naa siya sa mitochondrial matrix. Pero ang imong ETC, guys, kinaa siya diri sa inner mitochondrial membrane. Okay? So Yeah, na ay compartmentalization. So, diri nato makit-an si pyruvate oxidation and Krebs cycle and etc. Then sa cytoplasm si glycolysis. Okay, glycolysis. So, glycolysis can occur without oxygen. Okay? So, that's why anaerobic ni siya, guys. Okay? Um, to make or how do you make glycolysis in its simplest term? Glycolysis is actually just the conversion of glucose into two molecules of pyruvate. Then na ma-produce nga 2 ATP na through substrate level phosphorylation. Then wala lang nabutang diri no, but you know nga naasa ma-produce nga duha ka NADH. So mao lang ni ang glycolysis. Una-unaon nimo gitunga lang ang glucose para mahimong duha nga pyruvate molecules. Okay. So this is your glucose. So, ang glucose man gud kay pila iyang carbon atom. Very important po guys no para mas masabtan nato ang discussion sa kaning nga topic. Ato ang una-unahon ang number of carbon atoms. Kay mao natong timan-an. So, ang glucose kay unom. 1, 2, 3, 4, 5, 6 kay di ba ang chemical formula sa glucose is C6H12O6. So 6 na carbon, 12 nga hydrogen atoms, then 6 nga oxygen. So 6 po nga carbon ang naa diri. So in the step 10, ang product sa step 10 or the final product of glycolysis is pyruvate, di ba? Pyruvate is actually just tulo ka carbon tulo ka carbons nga molecule. Okay? So actually, gikuha ra na siya sa one glucose molecule. So kung tungaon nimo ang isa ka glucose molecule, maka-produce kag duha nga pyruvate. So two pyruvates, this finishes the glycolysis. Kay ang isa ka pyruvate, di ba, tulo ka carbon. So therefore, tungaon ra nimo ang isa ka glucose molecule para maka-produce kag duha ka pyruvate. So, glycolysis literally means splitting of a 6 carbon glucose into 2 pyruvates, each having 3 carbons. So, gitunga ra gyud nimo ang isa ka glucose molecule. So, that's why it's called glycolysis. Glyco means sugar, lysis meaning breakdown. So, i-breakdown nimo ang glucose. Okay. Yes. So, the stage 1 of cellular respiration is glycolysis. So, this is the part of the cellular respiration that doesn't need any oxygen. But I told you that the remaining stages need oxygen na. So, yeah. Okay. So, mao ni akong na-discuss na. So, C6H12O6. So, 2 C3 molecules in the form of pyruvate or pyruvic acid. So, the same lang na guys ang pyruvic acid o pyruvate. Okay. Then, naatay ma-produce nga NADH. Okay? So, recall po ha kung unsa nga step sa glycolysis ang makaproduce tag NADH. So, later ako po ipakita. Although, na-memorize na ninyo ni. So, glycolysis occurs in the cytosol. Overall, glucose is converted to 2 pyruvate molecules. So, released energy is stored in a net yield of 2 ATP and 2 NADH molecules. So, occurs under both aerobic and anaerobic conditions. So, it's a 10-step metabolic pathway. Okay? Divided into 2 phases actually. So, the first phase, steps 1 to 5, requires energy investment. So, mag-invest ta og energy ani. So, mag-utilize tag ATP. So, as you can see here, nagamit ang ATP. So, na-convert siya into ADP and a molecule of phosphate, inorganic phosphate. Yeah. Asa nga steps guys. Sa step 1 o sa step 3. Okay, nagamit ta og duha ka ATP. So, 2 molecules of ATP are dephosphorylated. So, when you say dephosphorylated, gikwaan og phosphate group. So therefore, na-release ang energy from the ATP, so nag-invest ta og energy. So actually gitrap na siya sa fructose 1,6-bisphosphate, which is the product of step 3, and glucose 6-phosphate, which is the product of step 1. Okay? So the final products of the first phase of glycolysis are 2 molecules of G3P, or glyceraldehyde 3-phosphate. The second phase is step 6 to 10. This is the phase of energy payoff. So diri actually bawiun nato ang gi-invest nga energy. Okay? So in this particular phase, ang gross nga ATP production, when you say gross, totality, wala pa gi-minus ang liability. So katong liability nato, katong duha ka ATP nga gigamit sa first phase. Okay. So pila ba ang gross ATP production diri? Remember, pila ang na-produce nga G3P duha. Diba? So therefore, the second phase must occur twice. Okay, kay duha man nga G3P ang na-produce, and only a molecule of G3P undergoes the remaining steps, the second phase. So what about the other one? Diba, sa step 5, si DHAP, which is dihydroxyacetone phosphate, ma-produce, ma-convert siya into G3P. So therefore, duha ang ma-form nga G3P. So therefore, kaduha dapat nimo occur si second phase. And unsa nga step ang nakaproduce ng ATP? Step 7 and step 10. Okay? Daw bi kay kaduha man nimo occur sa kada isa ka glucose molecule. So therefore, pilang na-produce? Upat. Kung i-minus na to ang gi-invest na to nga energy dire, so ang net is duha na lang. So that's why the gross production is 4 ATP but the net is only 2. Okay, kaya nagamit man tag energy sa first phase. So overall, this is the chemical reaction. And then, the final product is of course 2 pyruvates and duha ka NADH. Okay, sige. Himay-himayon na to. Asa ta nakaproduce og NADH? Asa dapit? Dire sa step 6. Okay? So, yeah. Since kaduha man siya mo balik, then duha sad nga NADH ang ma-form. Okay? You might kanang kung ano, you might wonder, lailahi ang kanang kuan. Lailahi ang ang pangalan sa mga enzymes. For example, sa step 5, triose phosphate isomerase. But ang nabutang diri is isomerase lang. So okay ra na siya. Triose phosphate dehydrogenase or glucose 6-phosphate dehydrogenase. Sakto ba? No, dili. It's glyceraldehyde 3-phosphate dehydrogenase. Okay? So, kana siya nga enzyme, it can also be called as triose phosphate dehydrogenase. So, parehas ra na sila. Ang important lang, dili gyud mag-change kasi pyruvate kinase and si phosphofructokinase, because these are the enzymes, and apil po si hexokinase because these are the enzymes that have irreversible reactions. Okay, so ako na siya i-discuss later on sa regulation of metabolism. Nganong important ang mga enzymes that operate irreversible reactions. Okay. So, you remember ha, kung asa nga step dapit, nakaproduce og ATP, naka-reduce ta og NAD+, so nakaproduce tag NADH. Yeah. Then, asa sad nga steps sa first phase, nag-invest ta og energy. Okay, so that's just pretty much it. Okay, that's basically glycolysis. So, you really don't have to, kanang i-retain yun sa imong mind ang each step, kay I know nga malimtan ra gyud ninyo later on, but at least kabalo mo sa iyahang main purpose, which is to divide glucose into 2 3-carbon molecules, which is called pyruvate. So naay duha ka pyruvate nga molecule nga produce, and then na ay duhasad nga NADH nga ma-form. Okay, then ang the net ATP production, or this is the substrate level phosphorylation ni siya, guys. Okay, kaya nahitabo man siya sa glycolysis. So the substrate level phosphorylation, sa glycolysis makaproduce tag duha ka ATP nga net. Kani siya animation lang ni siya, no. Yeah. So ganan kayo kuani kuan nga animation. So gibutang na lang nako siya sa presentation, no. So sa step 1, na phosphorylate si glucose, so nakabuhat tag glucose 6-phosphate. So by the use of hexokinase, diba nga enzyme. Next, nag-invest tag ATP diri, so naka-produce tag ADP, and actually phosphate molecule, kay breakdown man nato ang phosphate bond sa ATP. Another ATP is used in the step 3, forming another ATP and forming another pi. Pi is actually just a phosphate group. Okay. Yeah. So sa step 6, nakaproduce ta og duha ka G3P molecules. Okay. Kay nagbutang man ta actually og phosphate group diri. Actually katong pi nga akong ingon, katong phosphate group mao nang gibutang sa, okay, balik ta ha. Yeah. So di ba nakaproduce ang ADP? So asa ang pi, asa ang phosphate group? Diri butang sa glucose 6-phosphate. Nganong gitawag siya nga 6-phosphate? Kaysa 6 carbon gi-attach ang phosphate group. So gikan na siya sa ATP. Okay? Kani nga phosphate group. Then remember nga gi-isomerize lang ang glucose 6-phosphate into fructose 6-phosphate. So same ra gyapon ang location sa phosphate group. Then sa step 3, nag-add og another phosphate group sa first carbon. So that's why ang tawag is fructose 1,6-bisphosphate, because a phosphate group is attached in the first carbon and then in the 6th carbon. Okay. Then the product of the step 5 is duha ka G3P. So gitunga lang ni. Okay. Para makaproduce og duha ka G3P. Next, sa step 6 na ay duha ka NADH nga na form. Okay? So kani gidungan na ni nga duha ka G3P, gidungan siya ni sulod sa second phase. So that's why automatic na nga duha ka NADH ang na-form. Sa step 7, 2 molecules of ATP are formed. Okay? Then in the final step, which is step 10, another 2 molecules of ADP are phosphorylated to form ATP. Okay. So, mao ni ang products nato. Duha ka NADH, upat ka ATP nga gross, but we have to pay off ang nagamit natong ATP diri. So, therefore, ang net lang is duha ka ATP. Okay. So, that is glycolysis. So, another representation of glycolysis is C6, this is glucose, and na pyruvate nga ma-form. Okay. Actually, these are glyceraldehyde 3-phosphate. Dili pa ma-produce si pyruvate. So, sa second phase, naka-produce tag duha ka NADH sa step 6. And upat ka ATP from step 7 and step 10. Okay, but mag-minus tag duha ATP. So that's why the net is only 2 ATP. Okay. Some single and double nucleotides have important biological functions. So, An example of this is your NAD, which is, the vitamin B3 derivative or the niacin derivative. So, of course, you know that the function of this NAD plus, okay, this is your NAD plus, is para ma-reduce siya, para butangan siya og electron, para mahimo siyang NADH, which is the reduced form. Okay? So, asa ang hydrogen nga gibutang? So, hydrogen, this is electron. Okay, electron na rin siya. So, this is from the food. So, ibutang na dira. So, duha na ang H diri. So, this is the reduced form. Kung i-oxidize ang NADH, may mo siyang NAD+. Okay? So, kaning naa sa step 6, guys, katong duha ka NADH, asan ni padulong? Sa ETC. Okay? Dili ra na siya mas stagnant diya sa cytoplasm. They have to be used kay kani siya is energy rich ni. So, kinahanglan i-harvest. So, bali siya lang ang mag-deliver og energy nga gikuha nato sa glucose from glycolysis. I-deliver na niya sa ATP para didto makaproduce tagdaghan nga ATP. Kay kanimang og glycolysis, it's not a very efficient way to harvest energy. Kay duha ra nga ATP ang nakuha sa isa ka glucose molecule. But dito sa oxidative phosphorylation, we can get as much as 32 ATP. So that's why your oxidative phosphorylation is a more efficient way to harvest energy from the foods that we eat. Okay. In the particular discussion, especially in the Krebs cycle, which ang intermediates manggod sa Krebs cycles, sa Krebs cycle kay mga organic acid. So that's why naatay mga suffix nga acid and ate, such as katong pyruvic acid and pyruvate. So they are actually similar. Okay? So in cells, ate and ic acid forms are in equilibrium. So it doesn't matter much which term we use. So that's why parehas ra na sila. So pyruvic acid, pyruvate, citric acid, citrate. So do not be confused with those suffixes. Okay, a little review of the mitochondrion. So yeah, in the electron under the microscope mo ni ang appearance sa mitochondrion. Kung drawing na siya, then inani. So remember that this is the matrix, this is the cristae kaning folds, then the outer membrane, and then the inner membrane. Okay. So what happens then to the pyruvate? Okay, di ba? Na-produce nga pyruvate dito sa glycolysis, dito sa cytosol. Then unsaon lugar ng pyruvate dayon? Unsa ang fate ana? Unsay metabolic fate ana? So actually the fate of pyruvate depends on oxygen availability. So when oxygen is present, pyruvate is oxidized to acetyl-CoA, which enters the Krebs cycle, and without oxygen, pyruvate is reduced in order to oxidize NADH back to NAD, forming lactate. So this is the pathway of fermentation, and this one is the pathway to Krebs cycle. So mao ni atong unahon kay cellular respiration man ni. So unahon nato ang padulong sa Krebs cycle nga metabolic route. So but before sa Krebs cycle natay ginatawag nga pre-Krebs. So when you say pre, di ba, before. So para before sa Krebs cycle dapat ma-oxidize pang pyruvate. So the product of pyruvate oxidation is actually acetyl-CoA. And remember guys nga ang first step ba sa Krebs cycle kay acetyl-CoA, di ba? Mag-combine siya with oxaloacetate to form citrate, di ba? So unsaon nato pag-produce og acetyl-CoA? I-oxidize na nato ang pyruvate nga gikan sa glycolysis. So connected ang glycolysis with Krebs cycle through pyruvate oxidation. So occurs in the mitochondria, okay? Then in the mitochondria guys, or mitochondrion, a multi-enzyme complex called pyruvate dehydrogenase. This has an acronym. Ayang acronym and is PDH. So PDH refers to pyruvate dehydrogenase. So this is the enzyme that catalyzes the reaction of pyruvate oxidation. So this is the enzyme that converts pyruvate to acetyl-CoA. So the products of pyruvate oxidation include 1 carbon dioxide, 1 NADH, and 1 acetyl-CoA, so which consists of 2 carbons from pyruvate attached to coenzyme A. So acetyl-CoA then proceeds to the Krebs cycle. So diri pud nga point no, dapat may baw-an nato kung pila ka carbon ang acetyl-CoA. Remember guys, ang pyruvate is C3, tulo yang carbon. Ang acetyl-CoA duha lang. So therefore, asa to na padulong ang isa ka carbon. So nganong duha na lang ang sa acetyl-CoA. Kay katong isa ka carbon gi-release as carbon dioxide. So bali, katong isa nga carbon from pyruvate gi-release as carbon dioxide. And the remaining two are formed into acetyl-CoA. Okay. So mauni ang kanang dagway ato no nga pathway which is the stage 2. So this is pyruvate. So tulo ka carbons. Musulod siya guys sa mitochondrion in the presence of oxygen. Okay, so musulod ra ni si pyruvate kung nay oxygen. So say for example nay oxygen. Okay? So maka-form tag acetyl-CoA kay gi-decarboxylate man nato. So this stage is also called as pyruvate decarboxylation because pyruvate is decarboxylated. So meaning gikuhaan siya og isa ka carbon para makaproduce og isa ka carbon dioxide. So kung nagkuha ta og isa ka carbon in the form of carbon dioxide, duha na lang nabilin nga carbon atom. So kana siya nga carbon backbone gibutangan nato og coenzyme A. So naka-form na dayon tag acetyl-CoA. Okay. So remember guys that coenzyme A is pantothenic acid derivative. So this is your vitamin B5. So in this stage no, naay interaction sa duha ka vitamins, vitamin B3 and vitamin B5 in the form of coenzyme A. Okay, so diri na gid ninyo mahibaw-an no nganong important ang mga B vitamins because they are much involved in cellular respiration. Okay. Okay, so human na to siya nga stage. So sa stage 3 na ta, which is the Krebs cycle proper or the citric acid cycle or the tricarboxylic acid cycle. By the way, the person that discovered Krebs cycle is Hans Krebs. Yeah, you have to blame him for discovering Krebs cycle. So that's why naatay Krebs cycle karon tungod niya kay na-discover niya. Okay. Yeah. But yeah, tungod pud ani niya daghan the knowledge of biochemistry really accelerated because of the discovery of this pathway. Okay. So in this stage of cellular respiration, the oxidation of glucose to carbon dioxide is completed. So this is why we exhale carbon dioxide. Okay. Okay, so diri actually guys nga metabolic pathway nga na completely oxidize ang glucose. So that means when you say na completely oxidize, tanan nga carbon atoms sa glucose, which is pila gani ka buok 6, na convert into carbon dioxide. So this is where glucose is completely combusted into carbon dioxide. So diri ta naka-produce og 6 nga carbon dioxide. Remember guys, di ba katong mga equation sa aerobic respiration natay ma-produce nga 6 water molecules out of glucose oxidation, oxidation of 1 glucose molecule. Then naatay ma-produce po nga unom ka carbon dioxide. So nakumpleto ang production of carbon dioxide from glucose diri sa Krebs cycle. Kani guys, sa stage 2, balik ta sa stage 2. Pila ang na-release nga carbon dioxide? Okay, but before that, we have to ask ourselves first and to remind ourselves again, pila ang na-produce nga pyruvate sa kada isa ka glucose? Di ba? Duha. Okay, duha ka pyruvate. So kaduha sad nimo balik. Kay duha man nga pyruvate ang na-produce sa kada isa ka glucose. So therefore duha sad nga carbon dioxide ang na cleave. So sa stage 2 pa lang naanay duha nga gikuha nga carbon atom gikan sa glucose para makaproduce og carbon dioxide. So the other four will actually be converted to carbon dioxide in specific steps in the Krebs cycle, which mao dapat pud ninyo maupod na ang inyong timan-an. So kung aha dapit sa Krebs cycle unsa nga step naka-release ta og carbon dioxide. So at this point guys, duha na nga carbon ang nakuha sa glucose. Okay. So atong abangan kung aha siya dapit nga step katong remaining nga 4 carbon ma-convert into carbon dioxide in the Krebs cycle. So the Krebs cycle oxidizes the acetyl group from pyruvate. So the acetyl-CoA. So i-oxidize ni Krebs cycle ang remaining carbon atoms in the pyruvate. Okay? So occurs in the matrix of the mitochondrial. So this is 8 steps. Okay. So the first step is ang acetyl-CoA nga na-produce nato sa na-produce nato sa pyruvate oxidation, i-combine nato with oxaloacetate. So 2 carbons ang acetyl-CoA, 4 ka carbons ang oxaloacetate. So therefore naka-produce ta og citric acid which is called citrate, which has 6 carbons. Okay? So that's why guys, ang tawag sa iyaha is citric acid cycle. It's because i-regenerate lang nato si citrate permi. Okay? So that's why Krebs cycle is also called as citric acid cycle. Why is it called tricarboxylic acid cycle? Because citrate has 3 carboxylic groups. So that's why ang tawag sa iya is tricarboxylic acid cycle. Okay, kani. This is the totality of Krebs cycle. So ang citrate nimo tulo na nga carboxylic group diha. So that's why citrate is also called tricarboxylic acid. Okay. So si acetyl-CoA nga gikan kay pyruvate i-combine with oxaloacetate para makaproduce tag citrate. And that is catalyzed by the enzyme citrate synthase. Then aconitase, dayon, then isocitrate dehydrogenase, then alpha-ketoglutarate dehydrogenase, succinyl-CoA synthetase, succinate dehydrogenase, fumarase, and finally malate dehydrogenase. So kani guys, they are called as intermediates, Krebs cycle intermediates, and these are organic acids. Okay? So that's why guys, kanang mga malate nga makita nato sa foods, like asa man nato makita ng malate nga unsa nga particular fruit ang taas og malate. Actually apple has high malate content. So kanang mga malate nga nakuha nato gikan sa foods, diretso na sila citric acid cycle. Dili na sila mo agi glycolysis. Diretso na sila i-feed sa citric acid cycle. So that's why malate among other organic acids also have calorie value. Kay pwede man sila makuhaan as a source of energy through direct feeding to the citric acid cycle. Okay. And very important pud at this point nga matiman-an nato ang pila ka carbon atoms ang mga intermediates sa Krebs cycle. So remember guys, ang citrate has 6 carbons. Kay nag-combine man ang oxaloacetate which is 4 carbons and acetyl-CoA which has 2 carbons. So kanus-a mahimong 5, then kanus-a na sad siya mahimong 4. Kay kinahanglan ba siya mo balik sa oxaloacetate. So kani si citrate mo balik ra nag-oxaloacetate. Okay? That's why it's a cycle. Kay it's just a cycle. As long as naay acetyl-CoA, citric acid cycle would operate again and again. Okay, basta naa lang acetyl-CoA. And asa na to ginakuha ang acetyl-CoA sa pyruvate? And asa na to ginakuha ang pyruvate? Mainly sa glucose. Okay. Sige, so dire matiman-an na to kung pila ka carbon. So pyruvate has 3 carbon, acetyl-CoA has 2 carbon atoms, citrate has 6 carbon atoms, isocitrate has 6 carbon atoms, alpha-ketoglutarate has 5 carbon atoms. Succinyl-CoA has 4 carbon atoms, succinate has 4 carbon atoms, fumarate has 4 carbon atoms, malate has 4 carbon atoms, and finally, oxaloacetate has 4 carbon atoms. So, asa dapit nga stage guys nga nag-change ang number sa carbon atoms diri sa step 3 and 4. So unsa gaining step 3? Conversion of isocitrate to alpha-ketoglutarate dehydrogenase, but by what enzyme? Unsa ang ga-catalyze ani nga enzyme? Unsa gani? Alpha-ketoglutarate dehydrogenase. So therefore, a carbon dioxide is released in this step. Okay? Then pag abot nimo sa step 4, another carbon dioxide is released. Unsa ni nga enzyme ang gakuan ani? Ang ga-catalyze ani? Succinyl-CoA synthetase. Okay? So, yeah. So, therefore, asa nga step sa Krebs cycle na completely oxidize ang glucose? Sa step 4. Kay diri nga step guys nga gikuha ang tanan nga carbon nga originally gikan sa glucose. Okay? Gi-transform siya into carbon dioxide. Then kana siya nga carbon dioxide ibutang na siya sa dugo, and asa na padulong carbon dioxide? Sa lungs. And through our lungs, unsa man ato na i-breathe out. So, kana nga carbon dioxide mao na ang gamiton sa plants para makaproduce og another nutrients. Yeah. So, diri sa step 4 sa Krebs cycle na completely combust ang glucose. Okay? Yeah, this is a very cute figure no kay na ay mga piggy banks. So, unsa man ning mga piggy banks? They represent the electron carriers. Kay mao ni sila ang moderatso sa ETC para makaproduce tagdaghan nga ATP didto. So, NAD plus is reduced to NADH in step 3. So, not just carbon dioxide ang na-produce diri, but also reduction of one NAD plus molecule into NADH. Another NAD plus is reduced to NADH in the step 4. Okay. Then, a molecule of FADH2 is produced or is reduced in unsa ni nga step 6. Then the last NADH is produced in the final step or in the 8th step of Krebs cycle. Okay? Asa ang substrate level phosphorylation dapit? Sa 5th step. Okay? So, sa 5th step, na-convert si ADP, actually GDP, then naka-form tag GTP. And kanang GTP, convert ra na into ATP. Okay? Sige. Guys, ganong times 2? Ganong na ay times 2? So, we have to ask ourselves again. Pila ganyan na form nga pyruvate sa kada isa ka glucose? Di ba duha? Okay? Then, pilang na-produce nga acetyl-CoA? Duha sab. Diba? So, therefore, kapila ni mo balik? Kaduha sad ni mo ran. Okay? Kay duha gud ka acetyl-CoA ang gamiton. So, therefore, kaduha pod ni mo balik. So, therefore, ang na-produce na nga carbon dioxide, times 2 na to na. So, naisa diri nga carbon dioxide, naisa diri. So, duha. So, times 2, 4. Pila ganitong na-produce diri nga carbon dioxide sa pyruvate oxidation sa stage 2 duha, di ba? So 2 plus 4 6. So kana siya nga 6 carbon, mauna siyang originally nga gikan sa glucose again. Okay. Pila nga NADH ang na-produce sa kada isa ka acetyl-CoA? Tulo. 1, 2, 3. So kaduha man mo ran, kay duha man nga acetyl-CoA. So 3 times 2, 6. So therefore, 6 ang NADH nga ang na-produce sa to sa Krebs cycle. Pila nga FADH2 ang na-produce? Isa. Kapila gani mo ran? Kaduha. So 2 times 1, 2. So therefore, duha nga FADH2 ang na-produce out of 1 glucose molecule. So this happens twice for each glucose molecule. So sa kada isa ka glucose molecule, kaduha ni mo ran. Okay? Okay. So citrate, alpha-ketoglutarate, succinyl-CoA, succinate, and this is actually malate, oxaloacetate. And oxaloacetate is combined with acetyl-CoA to form citrate. So, per 1 glucose molecule pila ang na-produce? Duha ka ATP. So, this is substrate level phosphorylation. 6 sa carbon dioxide. Bali, 4 sa Krebs cycle and duha sa pyruvate oxidation. Pila ka NADH? 6 from Krebs cycle, then duha nga NADH sa pyruvate oxidation. Remember, di ba, katong pyruvate oxidation na ay ma-produce nga NADH, duha ka buok. And pila na produce nga FADH2, duha. You also have to remember asa nga step na produce ang FADH2, asa nga step ang substrate level phosphorylation sa Krebs cycle. Okay? So you have to take note of that. And of course, kung asa dapit ang NADH na form. Okay. So, yeah. Ang dakong pangutana ani, unsaon lugar ning mga NADH? Remember guys, naapatay NADH dito nga nakuha sa glycolysis. Okay? O naapay duha ka NADH nga nakuha na to sa glycolysis. Plus 8. So at this point, naanatay 10 nga NADH. Kung i-combine na itong na-produce na to sa glycolysis and citric acid cycle. So the remaining steps of the Krebs cycle, release of 2 molecules of carbon dioxide, okay, reduce 3 NAD to 3 NADH, reduce 1 FAD to FADH2, then produce 1 ATP. So this is per acetyl-CoA. Since duha man ka acetyl-CoA, so times 2 ni. Okay? Then i-regenerate si oxaloacetate. I also have to remember guys nga always ma-regenerate si oxaloacetate. So actually ang gi-oxidize guys, ang gi-decarboxylate, ang gi-convert into carbon dioxide is just the acetyl-CoA. So ang acetyl-CoA bali sila kani nga mga intermediates ang ga-deliver lang sa acetyl-CoA, no? So pag abot diri si acetyl-CoA nawala, nahilis. Pag-abot sa step 4, nahilis si acetyl-CoA. So, nahilis siya into carbon dioxide. Then, ang na-produce kay 4 carbon atoms. Okay, so, remember that oxaloacetate is 4 carbon. So, at the end of each cycle, si oxaloacetate ma-regenerate. Nganong kinahanglan ma-regenerate si oxaloacetate? Para makaproduce ta og another citrate. So para maka-carry ta og another acetyl-CoA. Okay? Para ang citric acid cycle mo run again and again. So that's why very important nga ma-regenerate si oxaloacetate at the end. Okay? Yeah, another animation guys. Okay, ganan pud ko ani nga animation. Kani pyruvate, pagsulod ni pyruvate sa mitochondria. Okay. Okay, so a NADH molecule is produced, then a CoA is combined to acetate in order to form acetyl-CoA, and it is combined with oxaloacetate to form citrate, and step 2 na dayon. Okay. Step 3. Okay. Step 4. Remember ha, natay na produce ng carbon dioxide diri ug natay na reduce nga NADH ani. Then there is a step 5 actually naka-produce tag ATP, substrate level phosphorylation sa step 5. Then sa step 6 naka-reduce tag FADH2. Okay, yeah. Then in the last step, which is the 8th step, naka-produce tag another NADH. Yeah, di ba? So Krebs cycle is very very easy. So I hope at this point guys nasabtan na ninyo siya. Okay? Kay kinahanglan gyud di ay siya explain in simpler terms. So ang metabolism is quite overwhelming, especially if wala siya gi-explain. Okay, but if gi-explain siya tarong, dali ra man di ay siya. Okay, basta timan-an lang ninyo tong mga points of view nga akong gi-discuss. So, sige, let's do an accounting of what we have done so far. So, nahuman na lugar ang Krebs cycle. Okay? So, i-account na to ang mga nahimo na to. So, 6 na carbon dioxide, kay na completely combust na to ang isa ka glucose molecule. So, 2 of which kay gikan sa pyruvate oxidation. Gikan sa stage 2. So, the other 4 is produced in the Krebs cycle. 4 ATP, 2 of which comes from glycolysis. Then the other 2 is substrate level phosphorylation sa Krebs cycle, sa step 5 sa Krebs cycle. 10 NADH ang atong na-produce so far. So duha gikan sa glycolysis, then another 2 gikan sa stage 2 which is pyruvate oxidation, then the other 6 kay na-produce sa Krebs cycle mismo. And duha ka FADH2 nga na himo nato sa step 6 sa Krebs cycle. So ang NADH and FADH2 i-deliver na sila sa electron transport chain. Okay? Okay, so sa stage 3 na ta sa cellular respiration. So diri actually mosulod si oxygen. Okay? So di ba you might ask, asa dapit si oxygen mosulod? Wala, dili dito sa glycolysis, dili dito sa Krebs cycle. But dito pa siya sa ETC. So, diri ga kahitabo guys ang oxidative phosphorylation. So this is the process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to oxygen by a series of electron carriers. So the oxygen in this pathway serves as the final electron acceptor. So, mauni siya ang dagway ni electron transport chain nito. Kani siya guys, this is just the inner mitochondrial membrane. Okay? So, electron transport chain is a series of membrane-bound electron carriers. So, embedded siya sa mitochondrial inner membrane. Okay? So, you have to recall ha kung asat dapit tong inner membrane which na-discuss na ko ganina. Electrons from NADH and FADH2. So, kaning mga NADH and FADH2 nga nahimo nato from glycolysis and Krebs cycle, they are transferred to the complexes of the ETC. So, kaning complexes, these are just the proteins. Okay, nga nung gitawag siyang electron transport chain because this is a chain of proteins that serve as electron receivers. So, sila ang mag-receive sa electrons nga gikan sa NADH and FADH2. Okay, remember nga, nagbutang tag-electron sa NAD plus o FAD plus para ma-reduce sila into NADH and FADH2. So, now, i-unload na nila. So, meaning, i-kwaon na nila ang electron, i-donate na nila ang NADH and FADH2 sa mga proteins sa ETC. So each complex transfers the electrons to the next complex in the chain. As the electrons are transferred throughout the chain, some electron energy is lost with each transfer. So this energy is used to pump protons across the membrane from the matrix to the inner membrane space. Then a proton gradient is established. So the higher negative charge in the matrix attracts the protons back from the intermembrane space to the matrix, and the accumulation of protons in the intermembrane space drives protons into the matrix via diffusion. And most protons move back to the matrix through ATP synthase. And ATP synthase finally is the enzyme that uses the energy na nakuha nato dire sa proton force, motive force, ang kana siyang energy no atong gamiton para mag-synthesize ng ATP from ADP plus pi. Yeah. Di ba kani statements ra ni siya. Ako ni siya i-summarize later on kay naaman tay mga animations. So mas masabtan ninyo later. So this is the inner mitochondrial membrane. So kani diri, di ba? Diri naka-embed ang electron transport chain nimo. So the electron transport chain has 3 proteins. Complex 1, complex 2, and complex 3. Then the fourth complex, okay, which is diri gakahitabo ang oxidative phosphorylation. So diri actually ga-form og ATP sa ATP synthase. So this is the complex 4. Kani siya, this is ubiquinone. Ubiquinone is a semi-essential nutrient, di ba? Kato yung assignment. Participate siya actually in delivering electrons from the complex 1 to complex 2. Then si cytochrome C delivers electrons from complex 2 to complex 3. Then diri actually si oxygen gahulat sa complex 3. Then ang tanan nga electrons, di ba mag-deliver man ni siya electrons sa complex 3. So ibutang na niya sa oxygen. So nakaproduce sa daig water. So sa complex 3 nakaproduce og water. Then diri sad gahulat si oxygen because oxygen is the final electron acceptor. So kaning mga electron nga gidonate gikan sa NAD plus, then pad H2 actually diri sa complex 2, si oxygen ang mag-accept para makaproduce og water. Okay, kani animation para masabsabtan, no? Okay. The other name of complex 1 is NAD dehydrogenase. Then complex 2, the other name is BC complex. Then the third protein, the third complex, which is called cytochrome C oxidase complex. And the final complex in the ETC is ATP synthase. Asa si NADH gani na produce sa matrix, kay nga naman sa matrix man gaka-occur ang Krebs cycle. So katong mga NADH gikan sa Krebs cycle, then katong sa stage 2 sa pyruvate oxidation, and actually katong NADH no nga gikan sa cytoplasm i-deliver sad na padulong diri sa mitochondria. Remember nga si glycolysis na sa cytoplasm. So katong NADH nga na-produce dito i-deliver na pasulod sa matrix sa mitochondria. Okay? Okay, sige. Okay. Yeah. So, ang first step, guys. Okay. Ato sad ni una-unaon ha. This is the matrix sa mitochondria. This is the inner mitochondrial membrane. And this is the intermembrane space. And then, ang sunod ana is outer na. The outer membrane sa mitochondria. So, ato na timan-an nga mga structure kay essential na para masabtan nato ni fully ang ATC. So, si NADH, nagikan atong glycolysis and Krebs cycle, i-oxidize ni NAD dehydrogenase. So, kung ma-oxidize si NADH, mahimong NAD plus. Diba? So, asa na padulong si electron nimo? Okay. Okay. Si electron, i-transfer na actually. Okay. Okay. Wait. Sige. Ang gipakita lang diri guys no is ang pag-transfer sa hydrogen atom. Okay. Sige. So, since gikwaan man siya hydrogen atom, so gikwaan pud siya electron. So, kanang hydrogen atom, gipump na siya ni complex 1 into the intermembrane space. Si FADH2 guys, actually ma-oxidize siya sa sa complex 2, which is the BC complex. Si FADH2 ha, dili na siya diri sa complex 1, sa complex 2 dapat siya. Okay? This is actually connected with succinate dehydrogenase. So, sa kanipod nga portion no, na connect ang Krebs cycle sa ETC. Because this is also the enzyme, the same protein that catalyzes the step 6. Step 6 ba? Succinyl-CoA synthase. So, step 5. Okay, step 5. Okay. So, step 5 sa Krebs cycle. Okay. So, sakto ba? So, succinate dehydrogenase, fumarase, malate, or sa step 6. Step 6. Sorry, step 6 di ay. Succinate dehydrogenase, di ba ang step 6? Step 7 is fumarase. Naglibog na ko. Wait. Itong tron. Okay, so step 1, step 2, step 3, step 4, step 5, step 6. Yeah, step 6 ko dahil. Step 7, step 8, step 6. Okay. Sige, balik to dito. Okay. Sa step, kaning second complex sa ETC, naa diri si succinate dehydrogenase nimo. So, connected ang ETC sa step 6 sa Krebs cycle. So, that's why si FADH nga na-produce dito, diri sa deoxidize sa complex 2. Okay? Yeah, so another hydrogen is pumped. Then, yeah, another hydrogen is pumped here in the step 3. And diri maghulat si oxygen para si H plus ug si oxygen mag-combine into water. So, kung kakadaghan ang ang na-oxidize nga NADH ug FADH2, kakadaghan sad ang hydrogen nga ma-pump into the intermembrane space. So later on, mudaghan na kaayo ang hydrogen diri. So that's why naay ma-produce nga protein motive force or proton gradient. So kung mapuno na, kung concentrated ng intermembrane space o hydrogen atoms, kinahanglan si hydrogen atoms mubalik to the mitochondrial matrix. And to allow the re-entry of hydrogen atoms back to the mitochondrial matrix, moagi siya kay ATP synthase. So, yeah, di ba? So kanang mga hydrogen atoms nga na-concentrate diri kay asa man nagikan ang hydrogen kaning mga hydrogen atoms guys, gikan ni siya sa NADH ug FADH2. Okay? Sige. So kung kakadaghan ang na-oxidize nga NADH ug FADH2, mudaghan sad ang hydrogen atoms diri. So tungod ana no nga very concentrated og hydrogen atoms diri, kinahanglan na siya mubalik. Okay? Then imagine okay, para mas masabtan ni no, ato ni siya i-compare sa falls. Okay, remember this is the head of the falls. Waterfalls ba? Okay. 'Di ba hydroelectric power, hydroelectric plants, situated na siya usually sa mga waterfalls, di ba? Especially kanang Maria Cristina Falls. Actually na ay mga plants, di hydroelectric plants, then gina-convert ang uh, hydrostatic energy into electrical energy. So that's why the plants in the Maria Cristina Falls, they actually supply the electricity of Mindanao. Yes, kanang ato ang electricity, um part of that kay ginakuha nato sa Maria Cristina Falls. So imagine the amount of power, the amount of energy nga ma-harvest nato gikan sa Maria Cristina Falls. Okay? So the same pud ani. Mura siyag falls. So kumbaga, kaning hydrogen atoms, una-unaon ninyo mura siyag tubig sa falls nga mo fall down siya. So asa man na ginakuha ang energy sa Maria Cristina Falls, guys? Kanang sa moving water, kanang falling water. Okay? Imagine good kung naa ka sa base sa falls, then matagak ang tubig sa imuhang ulo, di ba? Matay gud ka diha kay grabe ang energy, grabe ang force ana. So ang energy nga ma-release sa falls gina-convert na into electrical energy by the plants nga naa didto, mga planta nga naa didto. Okay? So mao pud na, guys, ang purpose sa mga dams para pod makaproduce og kuryente gikan sa moving water. Okay? So parehas pod ani. So think of these hydrogen atoms as falling water. So si ATP synthase, bali mao ni siya ang dam, mao ni siya ang planta, mao ni siya ang machine nga ga-harness sa energy sa gikan sa pag-diffuse sa hydrogen back to the mitochondrial matrix. So that's why mao ni siya tong ingon dito, no, nga the accumulation of protons in the intermembrane space drives protons into the matrix via diffusion, and ATP synthase is a membrane-bound enzyme that uses the energy of the proton gradient to synthesize ATP from ADP plus pi. So grabe nga potential energy ang na-form diri. Daghan nga potential energy, and ang energy sa pagbalik gikan sa intermembrane space to mitochondrial matrix is harnessed by ATP synthase. So asa na niya dayon i-trap ang energy kay ADP para maka-form og ATP. So that's why an ATP molecule is produced. Kaya ang energy nga gi-harness niya gikan sa pag fall back sa protons, so these are protons man, i-harness na niya to form ATP. Okay, so I hope you got my analogy. Okay. Yeah, so naatay mga miniature o naatay mga minute nga mga unsa ni mga microscopic na mga machine sa ato ang lawas. And actually, guys, ATP synthase is probably one of the most efficient machines ever made. No man-made machine has matched the efficiency of ATP synthase. So some scientists say kay grabe gud ang pagka-efficient na ATP synthase as a machine. Okay? And this is actually one of the things in biochemistry, as what those scientists that believe in God, okay, they really cite kaning ATP synthase as one of the impossibilities nga dili daw ni siya ma-produce by evolution. Sa pagka-efficient sa ATP synthase, it needs a creator. Okay, mao nang ingon sa mga scientists that believe in God. Okay? So that's why mao na ang function ni ATP synthase nimo. Okay? So di ba? Kani nga mga hydrogen gikan ni siya sa foods that we eat. Okay? Then mao na atong gigamit para makaproduce tag energy. Okay. Guys, asa ang kanang kuan, ang electrons. Atoms man to, hydrogen atoms man to. Asa ang electrons kani diri. Okay? So mag-donate siya og electrons sa complex 1 si NADH, then i-transfer na siya into the chain, throughout the chain. Okay? So actually ang electron nga ang pagpasa sa electron no ga-add pud na siya og energy para later on makuha siya ni ATP synthase. So it's actually released by the it's actually captured by the proton gradient nga kanang makuan diri ma-form. Okay? Okay, so para mas masabtan no, napod ko ipakita nga video. Okay, wait. ETC. Yan ang simple. Katong okay, kani.
Professor Dave again. Let's take a look at oxidative phosphorylation.
He knows a lot about the science stuff. Professor Dave explains.
We've examined the first two steps of cellular respiration. These are glycolysis and the citric acid cycle. As it turns out, neither process generates much of an energy payoff, but the NADH and FADH2 that were generated in the citric acid cycle move on to oxidative phosphorylation, which generates by far the most ATP out of these pathways. This pathway utilizes an electron transport chain, which is a series of mitochondrial membrane proteins that sit in the inner membrane of the mitochondrion. And we refer to these as protein complexes 1 through 4. These proteins bear a variety of prosthetic groups, which are non-protein components that give the protein its functionality, including flavin mononucleotides and cytochromes. There is also one compound in the electron transport chain that is not a protein, and that's ubiquinone, a small hydrophobic molecule that is mobile within the membrane, and it is also known as coenzyme Q, or CoQ. Once NADH feeds electrons into the first component of complex 1, these proteins facilitate a series of redox reactions, shuttling the electrons downhill from one component to another, with each structure down the chain having a higher affinity for electrons than the last. This process does not generate ATP directly, but a byproduct of this series of electron transfers is the generation of a proton gradient across the membrane. Protons accumulate outside of the inner mitochondria. The proton gradient across not generate ATP, a higher affinity for electrons than the last. This process does not generate ATP directly, but a byproduct of this series of electron transfers is the generation of a proton gradient across the membrane. Protons accumulate outside of the inner mitochondrial membrane, which then go to power another protein complex called ATP synthase. As you might guess from the name, this is the component that synthesizes ATP. Because the proton concentration becomes greater in the intermembrane space than inside the mitochondrial matrix, the protons will spontaneously move with the gradient to re-enter the mitochondrial matrix, and the only route available is through ATP synthase. This process is called chemiosmosis, and because the gradient is able to do work through chemiosmosis, we also call this the proton motive force. And it is these protons that power ATP synthase in phosphorylating ADP to generate ATP. ATP synthase has a fascinating structure with a component that looks startlingly like a rotor, where individual protons can bind
so that's why
and cause it to spin
grabe
in
siya
such a way
ka-sophisticated
that
nga machine.
catalyzes phosphorylation of ADP,
Nanomachine.
kind of like a stream of water turning a water wheel, which can then power some other process. With the NADH and FADH2 that is generated from a single molecule of glucose, we can get around 26 or 28 ATPs from this activity. So this is the pathway that generates the majority of cellular energy. So in summary, your body uses a variety of metabolic pathways. Glycolysis, the most ancient, produces 2 ATPs per glucose. But it also results in pyruvate, which can enter the citric acid cycle. Here, those 2 pyruvates generate another 2 ATP, but they also generate 6 NADH and 2 FADH2 molecules that can go on to the electron transport chain, which gives us our big ATP payout. Feel free to use this summary of the 3 steps in cellular respiration if you need to remember the main points of each process, all starting with just 1 molecule of glucose. Other sources of food like proteins, fats, and other carbohydrates besides glucose are initially broken down in unique ways, but this will always result in some compound that is eventually fed into one of the pathways we discussed. So all the food we eat eventually gets broken down this way. This is why mitochondria are regarded as the engine of the cell, since most of the energy needed for cellular activity comes from them. So the next time you're walking home and you feel tired, just think of all those trillions of enzymes slaving away to help you move your feet. It might put a little pep in your step. Thanks for watching, guys.
Yeah. So, katupod no, kani important po ni siya nga katong ingon na ko nga carbohydrates lang sa ta. So the other pathways leading to the breakdown of proteins and fats, ma-breakdown ra na sila into acetyl-CoA. So all of them must be fed into the Krebs cycle para makaproduce og energy. So that's why your acetyl-CoA is called as the central metabolite, kay tanan nga macronutrients mag-converge sila into acetyl-CoA para makaproduce og energy. So all of them are to be fed into the TCA cycle. So that's why your TCA cycle is also called the central metabolic pathway.
Eventually gets broken down this way.
So yeah, okay. Ang kanilang the only problem that I got here is kulang og complex 4. So actually ang ATP synthase is the complex 5. I think this is the old version of the book. And actually dili pa gyud ni siya kanang it's still debatable kung pila ba gyud ang proteins ang involved sa ETC. Nay uban nga books tulo, nay uban nga books resources, just like we saw from this video. Upat daw kuno. Okay? So that's why your ATP synthase sometimes it's called the complex 4 or the complex 5. But yeah, we might call this as the ATP synthase na lang. Okay, so tungod atong mga electrons nga gikuha nato from the oxidation of NADH and FADH2, naka-generate tag protein motif 4. So sa pagbalhin sa mga electrons throughout the chains, it also allows the pumping of hydrogen. So tungod sa pag-transport nato og electron from one complex to another, ang hydrogen atoms kay na pump to the intermembrane space. Then i-harness din siya sa ATP synthase like a machine that converts hydroelectric power into electrical energy. So your ATP synthase does the same. It actually powers the conversion of ADP and pi to ATP. So that's why guys, nganong gitawag siya nga oxidative phosphorylation. It's because kinahanglan siya og oxygen para mas ma-phosphorylate si ADP into ATP. So that's why if you compare substrate level and oxidative phosphorylation, mas daghan yun kang makuha nga ATP sa oxidative phosphorylation. So therefore, ETC generates most of the ATP from glycolysis. Okay. So that's why it's called na oxidative phosphorylation. So, kabalo na mong gitawag siyang oxidative phosphorylation. Okay. Yeah. So that's your ETC. Diba? So, if you can think of it, ang mga electrons nga gikan sa foods na to, mao day na ang mo power sa ATP synthase. Okay, kay tungod man sa pag-transfer sa electrons na pump man ang naka-generate man tag protein motive force. Okay, then ATP is an ADP is phosphorylated to ATP, and energy is energy is trapped to ATP through chemiosmosis. Okay. So kani siya guys, this is bioenergetics. So atong i-account no ang mga na-produce nato nga ATP. So you might also wonder no nganong gala ilahi ang mga number of ATP nga ma-produce. Okay? Depende sa source, but we have to we have to stick na lang to this kind of accounting of ATP para uniform ta. Then according to my readings, mausad pud ni ang ginagamit sa majority. So actually ang ma-produce sa kada isa ka glucose molecule is 32 ATP. Okay, nga naman. Sige. NADH is from one molecule of NADH makaproduce tag 2.5 ATP. Then from 1 FADH2 makaproduce tag 1.5 ATP. So katong action dito sa NADH and FADH2 makaproduce og inani nga amounts of ATP. So since naatay na produce nga duha ka NADH sa glycolysis, then duha ka NADH from pyruvate oxidation, and 6 nga NADH from Krebs cycle, so pila ka NADH tanan? 10. Pila ang na-produce na tong ATP from substrate level phosphorylation? 4 lang. Pila ka FADH2? Duha. So 10 times 2.5, 25 ATP. 2 times 1.5, 3 ATP. So 25 plus 3, 28 ATP ang na-produce sa oxidative phosphorylation. Then 4 nga ATP ang na-produce sa substrate level phosphorylation. So 4 plus 28, a grand total of 32. Okay? So mao ni atong i-follow nga system of accounting of kuan. Although it doesn't mean nga sayop tong 28 nga gikan sa video, then some books kay mag-ingon nga 38. Nganong nahimuan to 38 kay some authors consider kanang 3 ATP ang ma-produce sa NADH, then 2 ATP sa FADH2. Pero nganong naay 0.5 guys? Gimimik lang kung unsa gyud ang actual nga mahitabo. So dili daw kuno 38 gyud. Some say 32 ra daw. So that's why inani ang formula nga gigamit. Okay. So this is just theoretical yield, but we have to stick to this one para mas simple, no? Then yeah. This is very important ha, nga may baw-an nato ni kay mag-account pud ta sa pila ka ATP ang na-produce. So this is from one glucose molecule. Pila ka atong ma-produce nga ATP gikan sa isa ka fat molecule. Okay? Sa isa ka palmitic acid, for example, which is a fatty acid. So mao na atong i-discuss sa fat catabolism. Okay. The net yield, kani kay lahi. Wala na ako na-edit, no, but lagi 32 or 34. So 6 water are formed when the electrons unite with oxygen at the end of the electron transport chain. So we breathe because we need oxygen as the final electron acceptor. So the resulting ATP is able to leave the mitochondria by the ATP transport protein in the membrane, so it goes wherever it is needed in the cell. So we breathe in because we need oxygen. Because they are the final electron acceptor. So as what we have seen here, na asya diri sa ends electron transport chain. I have to clarify, ATP synthase is not a part of the ETC. It's actually a separate protein. So that's why mas proper gyud nga dili nato siya i-label as complex 4 or complex 5. It's just ATP synthase. Okay. So si oxygen muhulat siya sa fine sa end sa electron transport chain, which is either sa third or kung ay ikaupat, then sa fourth. Okay. So, what would happen guys if magkukulang tag oxygen? ATC would not operate and oxidative phosphorylation would be hampered, and then it would lead to quick death as what you have learned from the previous discussion. So that's why malumos ta guys, dili ta kaginhawa. You know nga kinahanglan na to oxygen, kung wala tayong oxygen mamatay dayon ta. Kay nga naman, kay kinahanglan man nga makaproduce tag ATP, kay kung walay oxygen walay ATP nga ma-produce. Okay, kay dili mag-operate si ETC kung walay oxygen. So that's why we need to breathe oxygen. Okay. Yeah, kani this is for your self nga evaluation na lang kung yeah. I-edit ni siya guys ha. So ako na lang i-edit karon be. This is 2 ATP. Ay 2.5 ATP. Then kani 1.5 ATP. Yeah, although okay ra gyapon ang 3 ug 2 gamiton, but since mag-stick man ta atong 32 ATP, so then mauna po ang recommended sa ubang authors. So, yeah. Okay. So, to help you review, mauna inyong balik-balikan. Kay ako man ipasa po nga PowerPoint sa inyo ha. Okay. Okay, each NADH produces 3 ATP. Asa na sad to? 2.5 ATP. 1.5 ATP. So, kani, pila to? 32. Okay. Okay. Sige. So, actually, mao to ang cellular respiration, guys. Napatay other 3. Pero kani siya short na lang. Okay, so mahuman ra ta ani. By the way, I'm recording this at 1:53 a.m. My God, okay, but I have to finish this one. Okay, kasi 65th slide na ta din. 86 slides man eh. Anyways, kung walay oxygen, guys, ang pyruvate, dili siya ma-convert into acetyl-CoA. So, dili siya mosulod sa mitochondrion. So, kung anaerobic ang environment, ang pyruvate ma-ferment into lactic acid or alcohol. So, in other organisms like bacteria, alcohol ilang ma-form from pyruvate. But in muscle cells, in our muscle cells, atong ma-form is lactic acid. And this happens when the Krebs cycle cannot occur due to lack of oxygen. So, lactic acid in muscle cells can cause muscle cramps. So, kanang pamaol, okay, kung naka-experience mo pamaol, it's actually due to the accumulation of lactic acid. Okay? So, pyruvate is reduced to lactate to regenerate NAD+. Okay? Later, ako ni siya explain, no? So, nganong magka-cramps ka, pamaol, it's because of the lactic acid. Kay nga naman, kay siguro intense kayo imuhang physical activity to the point nga medyo nagkulang kag oxygen, so some of the pyruvate is not converted to acetyl-CoA but instead fermented into lactate. Okay, so without oxygen, lactate. With oxygen, ang pyruvate ma-convert sa masulod siya Krebs cycle, ma-convert siya acetyl-CoA. But without oxygen, sa muscle cells, ma-convert siya ito lactate. Then, in microbes, ang pyruvate ma-convert into ethanol. So, actually, mauni ang ginakuan. Mauni ang para makahimo tag alcohol, no? Okay, so that's why alcoholic fermentation. So, that's why makaproduce tag mga alcoholic drinks tungod kay gina-utilize na to ang metabolic capacity sa yeast and bacteria to form alcohol from pyruvate. So that's why ginabutang ni siya sa mga barrels nga walay oxygen para ang pyruvate ma-convert into alcohol. Kay mahimo ra man ni nga alcohol in the absence of oxygen. So that's why it's called anaerobic. But in humans, since dili man ta microbes, so dili ta makaproduce og alcohol because we know that alcohol is toxic to us. So kung walay oxygen, magkulang og oxygen ato ang cells, ang pyruvate ma-convert lang into lactate. Okay, so yeah. Okay, so the pyruvate is converted to lactate. Okay. Okay. Sige. So mao ni ang fermentation, guys. Kani sa alcohol man ni. So diri man ta since we are dealing with human cells man. So kani. So glycolysis, remember na iduha ka pyruvate, but without oxygen dili siya ma-form into acetyl-CoA. So instead ma-convert siya into lactate. So para maka-produce tag lactate si NADH. Remember si NADH ma-produce siya sa step 6, di ba? Then kani si NADH, since wala may oxygen, so dili sad ni siya mosulod sa mitochondria. Okay, wala good oxygen. So si NADH would instead be oxidized into NAD+. So para i-oxidize niya, ma-oxidize si NADH, i-reduce niya si pyruvate. So NADH reduces pyruvate to form lactate. And as a consequence, si NADH mahimong oxidized form, which is NAD+. So this is essential para si NAD+ magamit sa step 6. You know that step 6 needs NAD+ para naa siya ika-donate nga electron sa NAD+ para naay ma-produce nga NADH. Actually guys, katong sa kuan sa matrix sa electron transport chain, di ba na oxidize man dito si NADH, so naatay na form og NAD+ sa complex 1 ug complex 2. Sa complex 2 FAD+ nga oxidized. So kana sila, dapat mo balik na sila sa Krebs cycle. Okay, kay dili man mag-operate ang katong mga steps nga ga-require og NAD+ or FAD+ kung wala sila. So that's why after they are oxidized in the electron transport chain, they must go back to the Krebs cycle para magpadayon sa ang Krebs cycle. Okay? So same sa glycolysis. Ang step para mapadayon tong step 6, kinahanglan niya na ay NAD+. Okay? So to sustain lactate fermentation, NAD+ must be regenerated. Okay? Yeah, that's basically it. That's fermentation. Pila ang na-produce nga ATP? It's just 2. So that's why fermentation is an inefficient method of harvesting energy. So dili kayo siya efficient. Unlike oxidative phosphorylation nga 32 ATP mo ma-produce. But anaerobic respiration would only give you 2 ATP. So it's very small compared to oxidative phosphorylation. So that's why favorable nga naay oxygen. Kay mas daghan ma-harvest nga energy kung naay oxygen. Okay. So this is just a self-test. So I will leave this to your own kanang kuan. To your own, to your own evaluation. Okay? Kay naapod ko ihatag actually nung kanang assignment after this kay para mansiran pud ninyo. Next is the Cori cycle. So the Cori cycle, guys, remember katong mga lactate nga na-produce diri, kinahanglan ni siya i-recycle to glucose. So the Cori cycle does that para makabali para ma-recycle si lactate into glucose. So Cori cycle is the metabolic pathway that recycles lactate back to glucose. Okay? Sige, how does the corecycle do that? So, katong lactate nga na-produce, gikan sa muscle, ipagawas na into blood, then, ang lactate sa blood, i-deliver na niya sa liver. Because the liver is the only one capable of producing glucose from lactate. So, lactate is oxidized to pyruvate, and pyruvate through gluconeogenesis, it converts pyruvate back to glucose. Okay? So, that's why glucose is recycled. So, thanks to co-recycle, ma-recycle lang si lactate. So, dili ra masayang si lactate. Kaya ibalik siya sa glucose. So, once nakaproduce tag glucose balik sa liver out of lactate, so, ibalik na sad na sa muscle. Okay? Para ma-recycle si glucose. So, lactate from active muscle is converted to glucose in the liver. Okay? So, bear in mind nga, different nga tissues ang involved sa co-recycle. So, generation of lactate sa muscles, but production of glucose from lactate is done in the liver. Okay, so that's the co-recycle. So, thanks to this kanang kuan couple, no, they were awarded a Nobel Prize due to the discovery of the co-recycle. So, for their discovery of the course of the catalytic conversion of glycogen. So, that's why co-recycle is named as such kay sila ang nakadiscover ana nga metabolic pathway. Okay. So, yeah, at least na-recycle ra balik si glucose from lactate. So, wala ra na sayang si lactate. But, unsa pa'y mga other ways para maka-produce tag glucose? So, dili tanan nga glucose ang ginagamit sa lawas kay gikan sa foods. Pero technically, gikan yun sila sa foods in the first place. Pero naatay mga glycogen stores because you know nga kung in times of kanang abundant tag pagkaon, then taas atong food intake, so the excess sugar is converted to glycogen. But in times when we need glucose, so glycogen can be mobilized. Glycogen can be broken down to glucose. So that metabolic pathway is called glycogenolysis. So glycogenolysis is just the breakdown of glycogen to glucose. Okay? And you know that glycogen is stored in the liver and muscle. So this is the liver, guys, and this is the liver cell, and kaning mga granules, kaning mga dots nga black, these are glycogen granules. So only the liver and then the muscle cells are able to produce glycogen from excess glucose. Okay, so this is the metabolic pathway of glycogenolysis. So glycogen phosphorylase breaks down glycogen into glucose 1-phosphate, and phosphoglucomutase catalyzes the conversion of glucose 1-phosphate to glucose 6-phosphate. Remember that G6P is the first substrate in the step 1 of glycolysis. So therefore, glucose 6-phosphate can come from glycogen directly. Okay? Yeah, so si glycogen ma-convert siya into glucose 6-phosphate that is fed into the glycolysis, and they can be completely oxidized through the Krebs cycle, or kung walay oxygen then to lactate. Okay, but remember that lactate can be converted back to glucose through co-recycle. So, di ba, na-integrate na nato ang mga metabolic pathways, glycogenolysis, okay, glycolysis, Krebs cycle, oxidative phosphorylation, fermentation, and para si lactate ma-convert balik to glucose 6-phosphate, kinahanglan og co-recycle. Okay. Yeah. So, Ang timan-an ninyo no nga mga enzyme kay si glycogen phosphorylase and si phosphoglucomutase. Okay? Si glycogen phosphorylase is actually the rate limiting enzyme involved in the glycogenolysis. So, unsa na mga rate limiting enzymes? I-discuss na nato na later on sa metabolic regulation. Glycogen phosphorylase is also the enzyme that needs pyridoxal phosphate as the coenzyme. Remember that pyridoxal phosphate is vitamin B6. Okay, so para lugar ma-perform ni glycogen phosphorylase iyang function, kinahanglan og PLP or kinahanglan og B6. So this is where B6 plays its role. So ga-play siya og role in glycogen breakdown. Okay, so B6 is required for the mobilization of glucose from glycogen. So remember guys, those B vitamins that we have discussed sa katong midterm. So nakit-an na gyud nato kung asa sila dapit. So si vitamin B6 sa glycogenolysis, si B3 tanan nga mga steps nga naay NADH. Si FADH2 tanan nga mga steps nga naay FADH2. And that is B2. NADH is B3. Asa si B5 as CoA. So yeah, diri siya sa acetyl-CoA. Diba? Asa guys si thiamine. Okay. Si thiamine naa siya sa PDH. Okay? PDH is the enzyme that is involved in pyruvate oxidation, katong stage 2 sa cellular respiration. So, para ma-convert si pyruvate into acetyl-CoA, di ba kinahanglan og pyruvate dehydrogenase. So, si TPP, thiamine pyrophosphate, okay, that's the coenzyme form of vitamin B1, di ba? So, in order for the PDH to function, it needs TPP as the coenzyme. Another enzyme that needs thiamine as the coenzyme is the alpha-ketoglutarate dehydrogenase. So, alpha-ketoglutarate dehydrogenase needs thiamine as its coenzyme. Okay. So, yeah. What about other sugars, guys, aside from glucose? Unsaon sila pag metabolize, unsaon sila pag catabolize? Actually, they can be fed into the glycolytic pathway. How? Fructose can be converted directly to fructose 1-phosphate. And fructose 1-phosphate can be cleaved into glyceraldehyde and dihydroxyacetone phosphate. And remember, si dihydroxyacetone phosphate naa siya sa step 5, di ba? Siya ang result sa step 4 sa glycolysis, and si DHAP, si dihydroxyacetone phosphate, ma-convert into G3P sa step 5, di ba? Sa glycolysis. And kung i-cleave nimo si fructose 1-phosphate, dili lang si DHAP ang ma-produce but also si glyceraldehyde. So through the action of trioskinase, si glyceraldehyde is converted to glyceraldehyde 3-phosphate. So this is how we feed fructose. Asa siya dapit mo enter? Sa step 4. I know, sa step 5. Okay, sa step 5 siya mo enter. Okay. Kaya dito man nakaproduce ta og G3P. What about galactose? Galactose can be converted directly to glucose 6-phosphate. So sa step 1 or sa step 2 siya mosulod si galactose. Okay? Then another enzyme that can cleave fructose to fructose 6-phosphate directly is hexokinase. So hexokinase can also phosphorylate fructose directly para makaproduce og fructose 6-phosphate, which is the third step of the product of the second step. So therefore, fructose can directly enter the glycolysis via step 3. Okay. And katong si fructose ng isa, So sa adipose tissue, sa adipocytes, si fructose ma-convert into fructose 6-phosphate. But in the liver, it's only in the liver no nga makaproduce tag DHAP and G3P out of the fructose. Okay. So, this is how the galactose and fructose enter the glycolytic pathway. So, sa glycolysis sila mosulod. So, in summary, okay, si glycogen, okay, i-break down into G1P and then G6P. Si fructose, either dito sa fructose 6-phosphate by the action of hexokinase or by the action of fructokinase, dito siya sa stage as a step 5. Mannose, guys, can be converted to mannose 6-phosphate and then to fructose 6-phosphate. So, musulod siya sa step 3, ang mannose. So, mannose is another monosaccharide. It's another sugar. Lactose, you know nga, kinahanglan nga i-digest siya ni lactase to form glucose and galactose. Trehalose can be converted into 2 glucose units by the enzyme trehalase nga makitaan sa small intestines, di ba? Yeah, so this is how we metabolize the monosaccharides, the sugars. They are all fed into the glycolysis at different points of glycolysis. So you take note ha kung asa sila dapit mosulod. Okay, so that's the end of our topic for catabolism of carbohydrates. Okay, so for the catabolism of proteins and fats, we will discuss that in the other video discussion. So at this point, nahuman na ning tanaw ang atong video discussion and I know na amoy mga questions, no, so that's why we will have an open forum on Tuesday para maansera na ko tanan ning mga questions. But for now, thank you for listening and see you on Tuesday. Bye.